Bio Enzymes Part II will focus on the structure and function of enzymes. The essential idea is that enzymes control the metabolism of a cell. Here is the outline of topics for the 3 parts of the enzyme unit. Use this slide to find your way to the section you need. This movie is focused here. Enzymes are globular proteins that lower the activation energy of a reaction, thus accelerating the chemical reactions in living organisms. Enzymes are relatively large as compared to the molecules on which they act. And enzymes have an active site, the location on the enzyme, where the substrate binds and undergoes a chemical reaction. Here are the first 3 important IB syllabus statements to kick off this movie. Define enzyme and active site. List 4 characteristics of enzymes. Describe the active site of enzymes. Again, enzymes are globular proteins that accelerate chemical reactions without being a reactant or a product, without altering the free energy change of the reaction. Enzymes have an active site for binding the substrate, the molecule or molecules on which the enzymes act. We'll get into that active site, and the enzyme brings those molecules to the transition state where either bonds will form or bonds will break. Enzyme catalysis involves molecular motion and the random collision of substrates with the active site. In this diagram, we can see the globular shape of the enzyme. Remember that the tertiary shape of the polypeptide is not random, but is quite precise based on the interactions of R groups that hold the tertiary shape of the enzyme. The overall tertiary shape of this enzyme has a region known as the active site that binds the substrate, which is the molecule or molecules on which the enzyme acts. The substrate joins the enzyme's active site through random collisions. Now, the complex shape of the active site results in certain amino acids with their unique R groups positioned in the active site, ready to bind the substrate. For example, if this substrate were a polar molecule, the R groups of these amino acids would also be polar. If the substrate were nonpolar, the R groups of these amino acids would be nonpolar. There is a strong specificity between the enzyme's active site and the substrate. And thus the lead-in to the next IB syllabus statement. Explain enzyme substrate specificity. Maybe you can already address this statement. In this diagram, the enzyme is on the right, and what we see here is nothing more than the active site, quite magnified. The substrate is bound to the active site. Specific R groups are positioned in the active site as a result of the tertiary shape of the polypeptide, the enzyme, which is dependent on the primary structure of the polypeptide, which is dependent on the sequence of bases in the genes of the DNA that would direct the synthesis of this enzyme. Now, the substrate joins the enzyme through the random collision of the substrate with the active site. The shape of the active site and the precise positioning of the R groups provides for the enzyme substrate specificity. Enzyme substrate specificity is important to the proper functioning of the enzyme to accelerate the reaction. Molecules whose shape prevents proper binding at the active site, as you can see here and here, will not be catalyzed. Here's a chance to review or practice what you know. Correctly identify the lettered structures in the diagram. A is the active site of the enzyme, letter D. B would be the substrate, substrates plural, about to enter the active site. You can see the enzyme substrate complex at E. The enzyme has positioned the substrates close together, facilitating the formation of a chemical bond. This is how the enzyme lowers the activation energy of the reaction. C is the product of the reaction, and the enzyme F is ready to repeat the process. The old model of the enzyme-substrate relationship is that of a lock and key. The lock and key model emphasizes the rigid nature of the active site as it binds the substrate. A new model, known as the induced fit model, has pushed the lock and key model aside. In the induced fit model of enzyme-substrate relationships, the enzyme undergoes a conformational shape change as it acts on the substrate. In this way, the enzyme brings substrates closer together or stresses the chemical bonds of the substrate, either of which lowers the activation energy required for the reaction to proceed. In this diagram, you can see the conformational shape change of the enzyme as the substrate enters the active site. In this diagram of the induced fit process, upon collision with the substrate, the enzyme undergoes a conformational shape change that serves to facilitate the catalysis of the reaction. In this diagram of an enzyme catalyzing a condensation reaction, an anabolic reaction, notice how the conformational shape change serves to facilitate the formation of the bond. This diagram once again displays a condensation reaction in which two amino acids are joined by an enzyme to form a dipeptide. The enzyme lowers the activation energy by bringing the two amino acids, the two substrates, close together to form a bond. Once the bond is formed, the dipeptide is released, and the enzyme is free to catalyze another reaction. This diagram displays a hydrolysis reaction. The enzyme, sucrase, binds the substrate, sucrose. Sucrose is a disaccharide. The enzyme lowers the activation energy by putting stress on the bond that holds the monosaccharides together, resulting in hydrolysis. Note the addition of water, hydrolysis of the disaccharide into monomers, glucose and fructose, which are released, and then the enzyme is free to work again. Now, hopefully this IB syllabus statement, which I showed you in movie 1, is clear. Explain that enzymes lower the activation energy of the chemical reactions that they catalyze. You can explain this by talking about the relationship between substrates and the active site. You know that enzymes lower the activation energy for a reaction to proceed. You now understand that the enzyme-substrate complex puts stress on the bonds of the substrate or brings substrates into close proximity. The induced fit of the enzyme to the substrate further stresses the bonds or further decreases the distance between reactants, again, both of which facilitate the reaction, accelerate the reaction. In this diagram of a free energy curve that clearly shows the lowered activation energy of an enzyme-catalyzed reaction, the reaction is a hydrolysis as a larger molecule, the reactant, is broken into two smaller molecules with the addition of water. The enzyme would stress the bonds that holds the molecule together, assisting with the breaking of the bond. This is what lowers the activation energy. The next part of this movie involves digging into this IB syllabus statement. Explain the effects of temperature, pH, and substrate concentration on enzyme activity. Draw the graphs that show the expected effects of these factors on enzyme activity. The factors that influence the rate of an enzyme-catalyzed reaction are temperature, pH, which is a measure of hydrogen ion concentration, and substrate concentration. Let's take a look at each separately. Pay close attention. Temperature influences the rate of an enzyme-catalyzed reaction. As the temperature increases, the reaction rate increases. When energy is added to a living system, molecules are moving faster, and faster-moving molecules have more collisions, and with more collisions between enzyme and substrate, there is an increase in the rate of the reaction. Now, at a certain temperature, approximately 37 degrees C, for this reaction curve, for this enzyme, the rate of reaction reaches a maximum. Temperatures above 37 degrees C results in lowered reaction rates. The rate of reaction declines dramatically with an increase in temperature above 37 degrees. At high temperatures, the tertiary shape of the enzyme is disrupted. The altered shape of the enzyme does not allow the proper binding of enzyme to substrate, so the reaction rate declines. Now, the enzyme involved in the curve on the right displays the exact same pattern, but at higher temperatures. The enzyme involved in this reaction, producing this curve, is an enzyme found in organisms that live in hot springs. Here are two curves displaying enzyme-catalyzed reactions as a function of temperature. And I'm asking you questions. As temperature increases at lower temperatures, why does enzyme activity increase? With more energy in the system, molecules are moving faster, increasing the number of collisions between enzyme and substrate, and with more collisions between enzyme and substrate, the reaction rate increases. Now, at X temperature, enzyme activity reaches a maximum. Yes, at X temperature, enzyme activity reaches a maximum because molecular motion is high, yet the enzyme has retained its native conformation. The shape of the active site retains high specificity for the substrate, despite the increase in molecular motion with the increase in temperature. Now, explain why enzyme activity declines above temperature X. The tertiary shape of the enzyme has been disrupted due to the increased molecular motion. The active site no longer binds the substrate, so the rate of reaction declines above temperature X. The change in shape of the enzyme is known as denaturing. And here's the relevant IB syllabus statement. Explain how enzymes are denatured. High temperatures or changes in levels of acidity cause the tertiary shape of enzymes to be disrupted. At high temperatures or with changes in pH, the active site no longer precisely fits the substrate. Rates of reaction decline or stop altogether when enzymes are denatured. Here's an image of an enzyme denaturing. Denature is to cause the tertiary structure of the polypeptide, the protein, to unfold, as with heat, base, or acid, so that its original properties, especially its biological activity, are diminished or eliminated. High temperatures or changes in pH will denature proteins, denature enzymes. As the egg white becomes opaque during heating, the protein albumin is becoming denatured. pH influences enzyme activity. Here are reaction rate curves for two enzymes, pepsin and trypsin, that function optimally at two very different pH levels. Pepsin has its maximum reaction rate at pH 2, while trypsin has its maximum reaction rate at pH 8. At pH 2, for pepsin, the tertiary shape of the enzyme accommodates the substrate well. The reaction rate is at maximum. But with any changes in the concentration of hydrogen ions, change in the pH to lower or higher values, the enzyme becomes denatured. The active site no longer binds the substrate, and the reaction rate declines. Here are rates of reaction curves for two different enzymes that have their optimal rate of reaction at different pH levels. And I'm asking two questions. Explain the term optimal pH with respect to enzyme activity. At the optimal pH, where the reaction rate is maximum, the tertiary shape of the enzyme retains its specificity for the substrate. The concentration of hydrogen ions at the optimal pH maintains the active site in a conformation that is suitable for binding the substrate. The second question, explain why the enzyme activity declines at pH values higher and lower than the optimal pH value. At pH levels above and below the optimal pH, the new concentration of hydrogen ions, whether that be more hydrogen ions or less, causes the enzyme to denature. The enzyme shape is altered so that the active site no longer has specificity for the substrate, so that it no longer can bind the substrate. The last factor that influences the rate of an enzyme-catalyzed reaction is substrate concentration, and here we have a curve with rate of reaction on the y-axis and substrate concentration on the X. As substrate concentration increases, the rate of the reaction increases. As more and more substrate is made available to a group of enzymes, the reaction rate increases as more and more enzymes become engaged with the increasing quantity of substrate. But that's only up to a certain concentration of substrate. Once a certain substrate concentration is reached, the rate of the reaction plateaus or reaches a maximum. Adding more substrate above that certain concentration does not increase the rate of the reaction. The reason is that all the enzymes are fully engaged. More substrate cannot increase a reaction that is dependent on available enzymes. If all the enzymes are engaged at this concentration, the reaction would be proceeding as fast as possible, a maximum rate. The last topic of this movie touches this IB syllabus statement. Explain the benefits of immobilized enzyme use in industry. Immobilized enzymes are widely used in industry. Immobilized enzymes are enzymes whose movement is restricted by being attached to a surface or other material. Enzymes can be attached to glass or held within a gel or attached to a membrane. The use of immobilized enzymes allows enzyme concentration to be high, increasing the formation of product. The enzymes can be reused, saving money. Immobilized enzymes are resistant to denaturing over greater ranges of temperature and pH. Immobilized enzymes prevent enzymes from mixing with the product. Here's an example of an apparatus where the enzyme is immobilized in a gel or immobilized by being attached to inert beads. The substrate is poured through the column and transformed to product as it contacts the high concentration of enzymes, immobilized enzymes, inside the apparatus. Here's a relevant IB syllabus statement to the use of immobilized enzymes in industry. Explain the use of lactase in the production of lactose-free milk. Lactose is a disaccharide which is difficult for certain people to digest. Lactose is poured into the column with the immobilized lactase enzyme. Lactase transforms the disaccharide into glucose and galactose monomers through a hydrolysis reaction. The milk that results from the process is lactose-free and consumable by people who are lactose intolerant. Here's another slide displaying the process of producing lactose-free milk using immobilized lactase enzymes in a column. Pour the lactose milk over the beads of lactase held by these inert carriers. The attached enzymes covering the surface of each bead renders the milk lactose-free. Glucose concentration increases, and the lactose-free milk will not disrupt the digestive system of those who are lactose intolerant, and it's a bit sweeter. Lactase is produced by infants to digest mother's milk so that they can gain nutrition from the milk. However, shortly after childhood, the production of lactase ends for many people. Thus, digesting foods with lactose can be difficult. Gas, abdominal pain, and diarrhea are typical effects of lactose intolerance. Lactose-free milk benefits these people and alleviates any suffering from consuming milk. There are various benefits of lactose-free milk. One is that glucose and galactose are sweeter than lactose, so less sugar is required when making milkshakes or fruit yogurts. Lactose tends to crystallize during the making of ice cream, thus making ice cream less smooth. Glucose and galactose are more soluble than lactose, increasing the smooth texture of ice cream. And bacteria ferment glucose and galactose more readily, increasing the rate of production of cheese or yogurt. And here is the last IB syllabus statement of the movie. Design experiments to test the effect of temperature, pH, and substrate concentration on the activity of enzymes. Experiments can easily be set up to test the effect of temperature, pH, or substrate concentration on the rate of an enzyme-catalyzed reaction. In this photo, you can see the tubes have been set up with the enzyme catechol oxidase. Catechol oxidase oxidizes catechol, converting catechol to benzoquinone, which is brown in color. You can see the tubes at room temperature and at 40 degrees C are significantly more brown than the control tube or the tube set up at 100 degrees C. At 100 degrees C, all enzymes would be denatured. The tube at 0 degrees C only had small amounts of enzyme activity. What I've described in words here is supported by the graphical representation seen here. Temperature can be manipulated using a water bath for the purposes of examining the influence of temperature on enzyme-catalyzed reactions. In fact, water baths can be used to control or stabilize the temperature of reactions because water has a high specific heat. Water, due to its cohesive H bonds, resists temperature change. In this image, you can see potato cells that have the enzyme catalase. Catalase engages with the substrate hydrogen peroxide to catalyze the decomposition of the hydrogen peroxide with the production of oxygen. Oxygen volume collected over time is a measure of reaction rate. Experiments could be run where varying amounts of hydrogen peroxide, the substrate, are used to determine the effect of varying the amount of substrate on the rate of reaction. You can see the theoretical curve for this experiment right here. The reaction rate increases with added substrate until all the enzymes are engaged, and then no further increase in the substrate will change the reaction rate. In this image, you can see potato cells that have the enzyme catalase. Catalase engages with the substrate, hydrogen peroxide, to catalyze the decomposition of hydrogen peroxide with the production of oxygen. Oxygen volume over time is a measure of reaction rate. Experiments could be run where the pH of the solution is manipulated to determine the effect of varying the pH on the rate of reaction. You can see the theoretical reaction curve for this experiment. Enzymes function at an optimal pH, and pH changes away from the optimal result in enzymes being denatured, which lowers the reaction rate. And that brings us to the end of IB Bio Enzymes Part II. Part 3 will look more carefully at metabolic pathways.